Okay, everyone, this is structure and support of the cell. This is the second part where I'll pick up where I left off. It should go pretty quickly though, because most of the pre-discussion questions that were actually asked have to do with the material that we covered in the first section, and that's more with the structure of the um, cytoskeleton, looking at the cell membrane, and the different parts of the cell membrane. So we got through most of those PDQs. So this part's going to go pretty fast. And let me see if I can do this. Nope. So I think, let me go through here. I'm trying to see if I can go. Aha. Nope. Right down here at the end here. I know we were finishing the cell membrane. So I think we had just gone up. We talked about cholesterol. We talked about what is meant by integral proteins, those that go through, and about how proteins can kind of move within the cell membrane to disperse. And it depends on if they're integral, if they can go through all the way, or if they're surface or if they're surface proteins. We talked about the different types of receptors, and I believe we left off on glycoproteins, which brings us to, oops, let me go right here. Let's go right here with glycoproteins to pick up. Okay, so I believe I can go ahead now. So a glycoprotein is an integral protein that has a carbohydrate, a polysaccharide, that kind of projects out. It's almost like a little antenna, and what it's used for is for cell-to-cell recognition. They are kind of like an identity marker. And so the antenna analogy is actually a good analogy because it picks up the signal of other cells that are nearby. Glycoproteins are a complication for organ transplants. The question is why? Okay, think about those markers on it. If the carbohydrate is not specific to the individual that it's coming from, then what's going to happen is it's not going to recognize that cell, or that cell's not going to be recognized as part of the body. And so oftentimes that leads to organ rejection, if you've heard about that. And so when they're doing an organ transplant, not only are they looking at the actual cells and the organs themselves, but they have to make sure that that reception, that signaling capability is matching what is going to be received by the cells that are also going to be there. Now, outside the wall, we do have a cell wall and the extracellular matrix, which is basically the fluid around it. Cell wall is found in plant cells, not animal cells. And it's just a cross-linked structural polysaccharides. So again, we have the cellulose. Remember the celery analogy? Cellulose, that long stringy, it helps build it up. Fungal cells use chitin. I can't even talk today. Peptidoglycan is for bacterial cells. But know that these types of carbohydrates are sort of the structural support of the different cells. Cellulose and chitin are probably the ones that you're going to encounter. Chitin is also found in insects as well, though. Okay. So here's just a little bit of a close-up. You don't need to know the details of the structure. Don't worry about that. But just if you see cellulose, recognize it as in part of the cell wall of plants. It's structure and support only. Cell walls are non-living, no cell walls in animal-like cells. And why do you think that is? What would happen if there was cell walls here? So think about that a little bit, and maybe in your reading come up with it. But what would happen if we had cell walls in our cells? And if that's the case, why do plant cells have cell walls in the first place? The extracellular matrix is a network of connected proteins and molecules outside of the cell membrane of animal cells. And the whole function of this is to kind of anchor cells on and to work for cell communication. So cell communication is not something that you've studied in general biology. It's very complex. it's not just individual isolated cells that are found inside the body, but there's a whole complicated, complex procedure for how cells are able to communicate, recognize each other, and move around. Only animal cells have any major extracellular matrix. Okay. So between cells, you have different kinds of junctions that help allow materials to flow between cells. They're proteins that connect cells to other cells, and depending on the junction, a channel between the cells may exist. So you can transfer substances between the two. Some of them are used for anchorage and transport, and those are the open junctions. So let's look at that. Okay, plasmodesmata is a good example of that. So when you're looking at a microscope, and we will look at them when we look at onion cells, you can actually see cytoplasm streaming, or that cytoplasm moving between the cells, and that's a way to transport nutrients. And so plasmodesmata are these open channels that allow this to happen. So that's considered an open junction. There's gap junctions. They're open channels between animal cells. And so it's the same process, allows materials to freely move. Plasmodesmata, you can actually see, we'll actually look at an example of it though. We also have closed junctions in animals. We call them desmosomes. They are like little rivets that anchor cells to membranes and tissues. And so like if you're looking at muscles, for instance. And then we have tight junctions, cell-cell connections that make a waterproof seal. And so it acts more of a boundary. We'll talk a little bit about these junctions between the cells when we look at cellular communication. Okay, so if there's any questions, let me know. Let me know in class, send me an email. Again, most of what you need to know for this particular unit is up through the cell membranes. We'll talk a little bit about the cell junctions when we look at cell communication. We'll talk a little bit about cell walls when we look at plant biology and photosynthesis. We do a little bit of just general plant info. And so we'll cover those two topics then. So if there's any other questions, just let me know.